state. Now, as I said, Yamanaka's group, so this is that paper, Takahashi and Yamanaka, but there are lots of groups that did this, including Thompson, that I already mentioned with the embryonic stem cells, Yanish, who I mentioned with the nuclear transfer. So there are many, many labs that contributed to this and all published this in a very short order of time. Yamanaka happened to be the first to do it. He did it really beautifully, and so I think, you know, if they could give the Nobel Prize to many, they might have, but they chose not to. So one question that does arise, though, is you make cells and you have them growing in a culture dish, and they sure look like embryonic stem cells, but how do you prove that they're embryonic stem cells or that they have the same properties? So one test of that would be if you can, so here's a mouse. So if you can take these and make these, oh, one thing I forgot to say, sorry. Really important. So Yamanaka, when he made these cells, I said they're ES-like, he called them induced pluripotent stem cells, IPS cells, or sometimes iPSCs. So these IPS cells, now this is a huge field of research now, these IPS cells. And so you can now, how do you prove that these IPS cells are actually pluripotent like an embryonic stem cell? So if you take a mouse and you make, so here's a fibroblast again, and you make IPS cells. So this is exactly the technique that I described. You put in these 4 factors and you grow them. These orange cells now, let's call them orange, if you inject them into a normal blastocyst, which is just an embryo that's developing, the question is, can those cells, and they're color-coded here, but these color codes really just reflect that there are actually markers that allow scientists to distinguish the origin. So these cells that are orange would give rise